The handouts or review materials must be treated with utmost confidentiality. It shall be the responsibility of the person whose name appears therein that the handouts or review materials are not photocopied or in any way reproduced, shared, or lent to any person or disposed in any manner. Any handouts or review materials found in the possession of another person whose name does not appear therein shall be prima facie evidence of violation of RA 8293. Top-notch review materials are updated every 6 months based on the current trends and feedback. Please buy all recommended review books and other materials.
Good day, everyone. I'm Dr. Ronnie E. Baticolon, and I'll be your lecturer for biochemistry here at Top Notch Medical Board Prep. I finished Intermed, BS in Basic Medical Sciences, and Doctor of Medicine at the UP College of Medicine. Afterwards, I took my residency training in neurosurgery at the Philippine General Hospital. After finishing residency training, I underwent subspecialty training in pediatric neurosurgery at the Royal Children's Hospital in Melbourne, Australia. I also spent 6 weeks at the Cure Children's Hospital in Uganda, where I learned to treat hydrocephalus using endoscopic third ventriculostomy and choroid plexus coagulation. Currently, I'm undertaking my master's in global child health at the St. Jude Children's Research Hospital in Memphis, Tennessee. I'm Professor 2 in Anatomy at the College of Medicine, University of the Philippines, Manila. I'm also a consultant pediatric neurosurgeon at the Philippine General Hospital. And then I have several positions at the moment. I'm the Secretary of the Academy of Filipino Neurosurgeons. I'm the Communications Co-Chair of the International Society for Pediatric Neurosurgery. And finally, I am a member of the Global Neurosurgery section of the journal Neurosurgery, published by the Congress of Neurological Surgeons. Now, some of you might know that I'm also a writer. I published my first book of essays in 2019. It's called Some Days You Can't Save Them All. This is available from the University of the Philippines Press, and this is where I wrote. In here, you will read about my essays on medical school and neurosurgery training. If you ever want to say thank you, or if you this is one way, this is one way. Or if you have a copy of the book, I hope that I am able to sign it in person at some point when we do see each other. Please feel free to share it as well to others who might find it interesting. So, punta na tayo sa biochemistry. How do you study biochemistry for the board exam? So, this is how we will do things. So, during your lecture days, which is essentially now, when you're watching this video, all I want you to do is to listen. So, when I teach biochemistry for Top Notch, I teach biochemistry like you don't know anything. Parang wala kang natutunan in med school. No? So, some of you might actually find the pace a bit slow, especially those who did biochemistry as pre-med. But I hope you understand that I'm really accommodating for everyone. So, what I'd like you to do is just listen. If you want to, you can take down notes. Okay? But the more important thing is that you try to absorb whatever you can. Okay? So, the focus of the first part of the lecture series is for you to just understand. Yun yung kasi yung mas important in biochemistry, because when you understand the pathways, how one concept relates to another, mas madali yung mag-memorize. Okay? So during the day, just listen, and then at night, you read again, okay? And then you answer the post-tests. Okay? So during the at this point during the lecture days, I really only expect you to memorize about half. Okay? About half of the material. I don't really expect you to memorize everything during the first round of listening or reading the lecture handouts. And then during the self-study period, this is when I expect you to read the handout again. So you read everything, and this is when you focus on memorizing. So hopefully by this time, you already have a good grasp of the metabolic pathways. So it will now be time for you to try to memorize as you will see later in the lectures, you don't really have to memorize everything. And I really just point out the most important concepts. Finally, during the crunch time, so this is when we have the live lecture. I will be going through the most important pathways with you. Again, you will have some practice tests. Uh, this is time when you just study the high yield topics again. So focus on the high yield topics. So you at this point you no longer have time to memorize, to go back and read everything again, no? Because you also have to study for your other subjects. So what are our expectations? So I expect you to read the self-study handout. So Dr. Rubio and I did prepare your handouts. We expect you to read them actually before you listen to the lectures. It would be nice if you already have read or at least scanned everything. Next, when I ask a question in class, please answer. So even if it's you're just at home or in the cafeteria or in the library, so you really need to answer. So the way I teach biochemistry is that every so often I keep asking questions, and what I am asking you to do, sometimes you might feel that the questions are repetitive, but that's really the key there, no? So repetition or retrieval of information, or periodic retrieval of information, that's what's been shown to be important, no, in long-term understanding and recall. Ah, so again, huwag niyo pansinin yung katabi niyo sa classroom or katabi niyo mag-review, no? So kunyari sabay kayong nanonood, no? So kapag nagtanong ako, no, even if you're in a small room, no, if you're studying with others, wag na kayong manghiyang sumagot, no? So I'd rather that you make the mistakes now than later when you actually take the exam, no? Than when you take the exam. Bawal mang-judge ng classmate, no? So ang mang-judge ng classmate babagsak ng exam. Okay? So mas important kasi na at least if you make the wrong answer, if you give the wrong answer, you already know the correct answer next time. Always remember that listening to the lectures is never enough, no? So unfortunately some students get the mistaken notion that all they have to do is listen to the lectures and that will be enough to pass the boards. So the board exam for biochemistry is really sometimes very unpredictable. The lectures, our goal here is not to teach you everything that will come out in the exam. We only cover about 80 to 85% of the board exam on average. No? So on average, meron talagang mga 10 to 20% na hindi natin alam kung saan nila kinuha. But over time, I've been teaching biochemistry in top notch for over 10 years now. And what I realize is that the more important thing that I'm able to impart to the students is that the students realize that they can actually learn biochemistry. And in fact, no, what you will be learning here from the lectures, I hope it's, even if for example hindi sila lalabas sa board exam, I hope they will become helpful when you do become physicians, no? Always remember the sequence is understand and then memorize. So you need to understand first before you memorize. Without understanding, it will be difficult for you to memorize. So I will teach you the frameworks, and from these frameworks, what I don't want you to depend on mnemonics alone, because if you depend on mnemonics alone, when you panic, wala na, hindi mo na siya masasagot. No? Finally, I hope you have fun. No? Finally, I hope you have fun. Biochemistry is the reason why I like teaching biochemistry is that it's not just memory work. It's not just about memorizing the origin, insertion, and action of muscles, or identifying the smallest parts of everything. No, I'm an anatomy professor, but really, the reason why I love biochemistry is that it's very much like mathematics. No, if you know the relationships of one pathway to another, if you understand what's happening, no, kahit pa ikot-ikutin yung questions, you will be able to answer them. No? I am on Twitter, so at Ronnie Bat. So I don't usually accept friend requests on Facebook. Aside from the fact that I don't really open Facebook much anymore, but I'm on Twitter and we can interact with that. No, so it's always nice to interact with students, especially now in the online setup or in the hybrid setup where we don't really get to see each other. So just to give you an idea, before when I started teaching biochemistry, we would have 2 or 3 full days of lectures. And when I started teaching biochemistry in top notch, there was a time when I was doing residency duties. So I would end up scheduling my lectures on Saturday afternoons from 12 noon to 10 p.m. Saturday and Sunday, just so I could be able to teach the students. To be honest, now it's as a consultant uh, and a teacher at the UP College of Medicine, it's becoming more and more difficult to try to find time to lecture to students taking the board exam. But I will try for as long as I can. No? I will try for as long as I can. I hope this is as fulfilling for me as it is for you. So this is going to be our outline. No? So Dr. Rubio, if you've listened to his lectures already, so Dr. Rubio discussed most of nutrition and for the structure of biomolecules. And now my role is for you to for me to try to make you understand how these four groups of biomolecules interact with one another. Okay, so first, we will be going through our first topic will be on bioenergetics. No, so in bioenergetics, we will learn about ATP, why you need ATP, how you use energy in the body, uh, and then we will discuss carbohydrates. So the carbohydrate pathways are main source of energy and also a storage form of energy. And then afterwards, we'll go to lipids. No, so lipids, uh, yan yung fats, triglycerides, cholesterol, lipid transport. No? So how they relate to diabetes and dyslipidemia. And then third group of biomolecules that we'll discuss will be the proteins. So we'll go through nitrogen metabolism. So I know you already discussed the structure of amino acids with Dr. Rubio, but here we will discuss how we actually metabolize them, no? And where they are actually useful for the body. After discussing those three main groups of biomolecules, we will be integrating them. So integration and nutrition, as well as vitamins and minerals. So uh, we will not just look at the structure of the vitamins and minerals, but we will now look at where the vitamins and minerals actually become part, no? Become part of your energy metabolism. And then there's a short lecture on important topics, no, special topics. So these are the complex molecules, your glycosaminoglycans, your glycolipids. Kasi may mga important diseases din dyan na lumalabas from time to time in the board exam. Finally, we'll go through nucleotides and genetics, the transfer of genetic information. We'll go back to the central dogma that I'm sure many of you already took up in pre-med, no? Many of you already took up in pre-med. So we'll look at the transfer of genetic information from DNA to RNA and then eventually to proteins. And then we'll give you an overview of genetic diseases, no? So and some technology some very brief overview of biotechnology. Okay? So once again, I hope, no? So I'm if there's anything that I'd like you to feel right now, so remember, take away the fear, no? I want you to feel excited because this is really fun. Our first topic is bioenergetics. So in bioenergetics, we will try to see why the body needs energy, how we use this energy. We will learn about ATP, how we produce it, and what happens to ATP. We will also learn what happens when we don't have ATP and why we don't want that. So some definition of terms first. What is bioenergetics? So bioenergetics is a transfer and utilization of energy in biologic systems. So you will see our body, no? So when we eat, we take in energy. And then when we take in energy, why do we take in energy? You know? So bakit ba tayo kumakain, di ba? We take in energy para makapagtrabaho tayo, no? Para magpag-exercise tayo, so we can do what we want, right? So, and then, we excrete, no? So, excess, yung mga hindi natin gagamitin, we excrete that. We also release energy. So, essentially, may pumapasok na energy sa atin and then may nire-release tayo na energy sa environment. So, there's this constant flow of energy. So, bioenergetics yung tawag doon, no? Now, some important concepts. When we say change in enthalpy, change to, kaya siya change in enthalpy or delta H. So delta meaning change. So what do you mean when we say enthalpy? Always remember, when we talk about enthalpy, we're mainly talking about heat only. So kaya pag change in enthalpy, change in heat. So delta H, change in heat between the content of the reactants and products. So what do we mean by that? So for example, ito yung reactants, ito yung products. Okay? So reactants, products in a chemical reaction. Kung ito yung heat content ng reactant, tapos ito yung heat content ng product, what happened? Did we gain or did we lose energy in the reaction? Okay? So, anong nangyari? Kung mas mataas siya sa reactant, mas mababa sa product, we lost energy, correct? So, in that case, no, that's the delta H or that change in enthalpy is positive. So, meaning to say, mas maraming energy yung reactant, mas maraming heat yung product, yung reactant kaysa sa product. So, delta H will be a negative. That will be what you will call an exothermic reaction. Kasi yung heat, nawala, no? So, saan napunta yung heat ng reactant? Napunta yun sa environment. That's an exothermic reaction. In contrast, kung ganito naman yung heat content ng reactant, ganito yung heat content ng product, what happened? Did you gain or did you lose heat? You gained heat. No? So, from reactant here, tumaas yung heat content niya, you gained heat. So, that's what you would call an endothermic reaction. You gain heat, endothermic reaction, therefore delta H or the change in enthalpy will be positive, negative, or zero. Positive kasi nag-gain ka ng heat. So this is change in enthalpy is measured in joules. So delta H, heat. The next concept is change in entropy. So when we say entropy, delta S. So change in entropy. S is entropy, delta change. So change in entropy is delta S. This is the measure of change in the randomness or disorder of the reactants and the products. Ibig sabihin, sabi nila, no, our universe is moving towards a state of entropy. Mas gusto daw natin mas disorderly. Diba? So, ibig sabihin, for example, before, when the universe was created, no, Big Bang, 'di ba? So simula sa isang solid, sa isang complex, no, very compact, very compact universe, no, na full of energy, dark energy, kung ano-ano pang energy, it's now rapidly expanding. Kasi yun nga yung mas preferred natin, no, yung higher entropy, no, mas disorderly. No, what about ice? So kunyari, if you compare ice with water, no, parehas naman niya may water molecules. Ice is solid, whereas water is liquid. Which one, ano nangyayari sa ice pag nilagay mo siya sa table? It will begin to melt. It will begin to melt kasi nga, bakit ang gusto natin solid to liquid? Kasi yung solid mas organized, yung liquid mas disorganized. So you're moving towards a state of higher entropy. So when you say change in entropy, delta S, no, it's a change in randomness or disorder. And what we prefer is higher entropy. So, paano ngayon papasok ngayon doon yung energy? So, doon papasok yung tinatawag natin na change in free energy or delta G. So, what is delta G? So, yung delta G, ito yung important na concept. Yung change in free energy, yun yung energy na available for us to do work. Meaning to say, kunyari ikaw, may gusto kang gawin, no? Gusto mong itaas yung kamay mo, or gusto mong mag-isip, or gusto mong sumipa ka. Kailangan meron kang free energy. Meron ka dapat delta G. Dapat meron kang delta G, no? So meron ka dapat change in free energy. Yung change in free energy, yun yung nagbibigay sa iyo ng energy to do work, no? So kino-compute natin yung delta G, no? We compute delta G using delta H and delta S. So the formula is here, no? Delta G equals delta H minus T delta S. So what's delta H again? Change in enthalpy. And change in enthalpy is change in heat content minus temperature in Kelvin times delta S or change in entropy. or the disorderliness, change in disorderliness or randomness of a system. So yan lang yung tatandaan nyo dito. The equation delta G equals delta H minus T delta S. So ano ngayon yung gusto natin? No? So delta G, it's a very energy available to do work. It is also the one that we use to predict whether a reaction will proceed spontaneously or not. So pag tinanong kita, itong reaction ba na to magpo-proceed or not? Itong reaction ba to magiging spontaneous or not? Anong sasabihin mo sa akin? Sir, I need to know the delta G. Okay, tatandaan nyo yan, ha? If you want to find out whether a reaction will proceed, whether a reaction will be spontaneous or not, you need to look at delta G. And to look at delta G, to compute delta G, what do you need? Delta H minus T delta S. So ano lang tatandaan nyo dito? Isa lang yung gusto natin. Ang gusto natin, for a reaction to proceed, for a reaction to be spontaneous and favorable, delta G has to be negative. Again, I'll repeat. For a reaction to proceed spontaneously, for a reaction to be spontaneous, for a reaction to move forward, for a reaction to be favorable, delta G has to be negative. Now, think about that, no? So, kung gusto natin na favorable yung delta G, so you will see here, if we want this, no, to be negative, anong kailangang mangyari sa delta H? Tumaas or bumaba? So, kung gusto natin ito negative, dapat yung delta H bumaba. No? So, in contrast, yung delta S naman, anong gusto natin mangyari kung gusto natin negative yung delta G? Dapat yung delta S, no? Dapat yung delta S tumaas. Okay, dapat yung delta S tumaas. Now, meron din tayong tinatawag na standard free energy change. So, when we say standard free energy change, delta G naught, this is delta G computed under standard conditions. So, meaning to say, when we say standard conditions, reactants and products are 1 mole each. The temperature is 298 Kelvin and the pressure is 1 atmosphere. Bakit kailangan natin ng delta G naught? We need delta G naught because, for example, if you want to compare enzymes, if you want to compare different enzymes or different reactions, they exist. The enzymes exist in different conditions. And so, if you want to be able to compare them, you need to standardize the conditions. Okay, so yun yung nagiging value ng delta G naught. So let's take a look at this graph. So when you look at this graph, okay, so let's try to see. Let us illustrate delta G. So you see here, no? So kanina sabi natin, ano ulit ang formula ng delta G? Delta G equals delta H minus T delta S. Change in enthalpy minus temperature in Kelvin times change in entropy. And anong gusto natin mangyari sa delta G para maging spontaneous and favorable? It should be negative. So to make delta G negative, kailangan ng delta H tumaas or bumaba? Bumaba. Pero yung delta S kailangan tumaas. But sometimes, no, hindi naman madaling makuha yung delta H and delta S. So there is one more way of computing delta G. So let's take a look at this graph. So you will see here, no? So you will see here, right? This is your substrate and this is your product. Okay, so this is your substrate on the left, this is your product on the right. The x-axis is time and the y-axis is energy level. Okay, so x-axis is time, y-axis is energy level. So if you compare the substrate and the product, you will see there, sino yung mas mataas yung energy? Sino yung mas mataas yung energy? Yung substrate or yung product? Yung substrate, di ba? Yung substrate. So therefore, no, pag tinignan natin, paano ba natin kukumpitin yung delta G? How do we get the change in free energy? To get the change in free energy, anong kukunin natin? Yung energy ng product minus yung energy ng substrate. Kasi gusto natin malaman, ano ba yung nangyaring change? Para lang yan nagko-compute ka ng weight mo. So gusto mong malaman, ano yung change in weight mo before you took the boards and after you pass the board exam, no? So gusto mo malaman ano ba yung change in weight ko, no, before I took the boards and after I passed the board exam, no? Of course, alam natin lahat 'yan magi-gain, no? No, but if you want to do that, what do you do, 'di ba? Di ba paano mo gagawin yung formula mo? Yung weight mo after you pass the board exam, isusubtract mo yung weight mo, no, before you took the boards. Parang ganoon din dito. To compute delta G, what do you need to do? Yung energy ng product, yun yung after, minus the energy of the substrate. So for example here, no? So delta G equals energy of the product, this one, minus energy of the substrate. So in this scenario, what happened? Did we gain or did we lose energy? So if you look at this equation, did we gain or did we lose energy? Okay. What happened here is that we lost energy. No? So you will see, ito yung substrate, tumaas, pero bubagsak. Ito yung final output na. Sa products, ito na lang. So you will see here, the change in free energy is positive, negative, or zero. Negative, no? So negative. So kung negative, no? So kung negative yung change in free energy, is this reaction spontaneous or not? It's spontaneous. Is this favorable or not? It's favorable, di ba? Sabi natin, basta delta G is negative, it's spontaneous, it's favorable. So let's compare, no? So for example, here. Okay, so look here at the graph on the left. And look here at the graph on the right. So let's compare the red graph with the blue graph. So in the red graph, you will see, no, look at the energy of the reactants and the energy of the products, no? So you will see there, dun sa energy ng reactants, no, dun sa red graph, no, mataas, mababa. So what happened? Did we gain or did we lose energy? We lost energy. So in that case, delta G is negative. Ang tawag dyan, exergonic reaction. So hindi na thermic, no? Kasi kanina, thermic kasi heat lang yung pinag-uusapan natin sa enthalpy. Now, we're talking about energy. Kaya exergonic, no? Exergonic, we release energy. The delta G is negative. And this reaction is spontaneous and favorable. Compare naman natin dun sa blue graph. When you look at the blue graph, ito yung energy ng reactant, ito yung energy ng product. What happened? Did we gain or did we lose energy? So, we gained energy. Pag kinumpit natin yung delta G, product minus reactant, it will be positive. So, that's what we call an endergonic reaction. Delta G is positive. Is this reaction spontaneous? No. Is this reaction favorable? No. Kasi nga, positive yung delta G. Okay. Paano kung no change? Kung no change, no, delta G is zero. In that case, the reaction will be in equilibrium. So the forward reaction happens at the same time as the backward reaction. And there is no change in energy. So, paano ba natin yun matatandaan? No? So, paano ba natin yun matatandaan? Ganito lang yan, no? Para ka lang nagsha-shopping, no? Paano na tayo nagsha-shopping? Ano ba yung mas madali? No? Ano ba yung mas madali? Yung gumastos o mag-ipon? Diba? O di syempre yung gumastos, no? So kaya yung paggastos, no? That's spontaneous and that's favorable. So gusto natin yung spontaneous. Okay, so always remember delta G is negative. So now, ibig sabihin ba nun lahat ng reaction sa body natin negative yung delta G? No? Ibig sabihin ba lahat ng chemical equation sa body natin delta G? Kasi nag-exist tayo, ibig sabihin lahat ng reactions gumagana. No, that's not true, no? So, hindi lahat ng reactions natin sa katawan negative yung delta G. No, merong mga positive yung delta G. So, paano nangyari yun, no? Meron kasi tayong tinatawag na concept called coupling reaction. So meaning to say, the delta G of two consecutive reactions are additive. Ibig sabihin, kung meron kang dalawang magkasunod na reaction, no, pwede mong ipag-add yung delta G nila, tapos malalaman mo kung mangyayari ba sila as a whole. So what do we mean by that? Let's take a look at this. So you see here two reactions. The top reaction is the conversion of glucose to glucose 6-phosphate. So you will see here, glucose combines with ATP to form glucose 6-phosphate. When you look at the delta G, so expressed here as delta G naught, parang standardized, no? When you look, the delta G is negative 4,000 calories per mole. Is this spontaneous? Yes. Is this favorable? Yes, because the delta G is negative. What kind of reaction is this? This is an exergonic reaction. You release energy. Now take a look at the second reaction. The second reaction is the conversion of glucose 6-phosphate to fructose 6-phosphate. This time, the delta G is positive 400. So 400 calories per mole. Is this second reaction favorable? No. Is this reaction spontaneous? No. So ordinarily, this reaction will not occur. Hindi siya mangyayari. No? Pero we do in fact have fructose 6-phosphate in the body. So, paano nangyari yon? Anong ginagawa natin? Sa yung body natin, pagsusunurin sila. No? Our body will have these reactions occur consecutively. Meaning to say, pagsasabayin natin sila, no? Pagsusunurin. So, what will happen is, if you add them up, ito yung magiging overall reaction. So, you will see here, glucose combines with ATP to form fructose 6-phosphate and ADP. So, pag pinag-combine natin sila, what happens? When we combine them, you will see yung delta G, ipapag-add natin. So, minus 4,000 plus 400, magiging minus 3,600 calories per mole. That's the overall delta G. And ibig sabihin, sinasabi natin sa coupling, Sinasabi natin sa coupling, yung overall delta G, yun yung magmamatter. So if you have two reactions, as pinag-add mo sila, both of them will proceed for as long as the overall delta G naught is negative. Again, the delta G's of consecutive reactions are additive. So kapag meron kang reaction 1, with a very negative delta G, pag sinabay mo siya with reaction 2 with a positive delta G, as long as the overall delta G is negative, both reactions will proceed. Ano ibig sabihin nun? Kunyari meron tayong pathway consisting of 10 steps. If you have a pathway consisting of 10 steps, okay lang na merong mga step na positive yung delta G. Bakit okay lang yun? As long as pag pinag-add mo yung delta G from 1 to 10, ang sagot dapat ay negative. Pag ang overall delta G ay negative, then the entire pathway will proceed. What else does this mean? So, for example, meron akong reaction. I have a reaction. Gustong-gusto ko talaga siyang gawin sa body ko. Unfortunately, the delta G is positive. Unfortunately, the delta G is positive. So, kung meron kang reaction sa body na gusto mong gawin, pero positive yung delta G niya, anong kailangan mong hanapin? Maghahanap ka ng reaction na ang delta G ay negative. Tapos, pagsusunurin or pagsasabayin mo sila. Is that clear? No? So, dun pumapasok si ATP or adenosine triphosphate. So, ATP or adenosine triphosphate, yun yung main role niya sa body. So, you will see this is ATP. When you look at ATP, so it's adenosine attached to ribose and 3-phosphate groups. So, kaya siya adenosine triphosphate. It's called the energy currency of the cell. So, siya yung energy currency of the cell natin. So, ano ibig sabihin? So, ginagamit natin siya siya yung nagbibigay ng energy. Siya yung nagbibigay ng energy. When you remove one phosphate, from ATP to yield adenosine diphosphate, what happens? Look at the delta G of that reaction. ATP converted to ADP plus phosphate, the delta G is negative 7,300 calories. Ibig sabihin nun, hydrolysis of ATP has a very negative delta G. So kung meron kang reaction na positive ang delta G, anong gagawin mo? Hahanap ka lang ng ATP, tapos sasabihin mo kay ATP, sabay ka naman sa akin. No? ATP, sabay ka naman sa akin. Mag-release ka naman ng phosphate para kahit positive yung delta G ko, magpo-proceed na din ako spontaneously. Okay? So, that's why, meaning to say, no? The transfer of energy from ATP makes it possible for you to have reactions proceed, no? Even though you have a unfavorable delta G sa beginning, no? So, let's take a look at this table. So, itong table na to, no, ang tawag ko dito, this is what I call the socio-economic classes of biochemistry, no? So, ito yung socio-economic classes of biochemistry. So, you will see here, so if I point out, look at ATP. Yung ATP nasa gitna. Okay? Yung ATP nasa gitna. So, you will see ATP, when you hydrolyze it, no? You release 7,300 calories. Okay? So, anong significance niyan? So, lahat ng nasa taas kay ATP, mas mayaman sa kanya. Lahat ng nasa taas ni ATP, mas mayaman sa kanya. So, you will see there, for example, phosphoenol pyruvate. Si phosphoenol pyruvate, if you remove phosphate, it releases 14,800 calories. Mas mayaman siya. Similarly, creatine phosphate. If you release phosphate from creatine phosphate, you will release 10,300 calories. Diba? Saan nyo narinig si creatine phosphate? Sa muscle physiology, sa exercise physiology, di ba? So, si creatine phosphate, no, ginagamit natin yan for rapid release of energy. So, yung mga yan, mas mayaman yan kay ATP. Mas marami silang ginagastos, no? Mas marami silang nailalabas na pera. Kaya sila nasa taas. In contrast, yung mga nasa baba naman, no? Yung mga nasa baba naman, these are the molecules which release a lower amount of energy when you hydrolyze them. So, when you look, glucose 1-phosphate, pag tinanggal natin yung phosphate, it only releases 5,000 calories. So, what does this imply, no? So, kunyari ikaw, wala ka ng pera. Kung wala ka ng pera, kanino ka mangungutang ng pera? Sa mas mahirap or sa mas mayaman? Siyempre dun sa mas mayaman, di ba? Doon ka sa mas mayaman manghihiram ng pera. In contrast, ikaw, pwede mo namang pahiramin ng pera yung mga mas mahirap sa'yo. No? So, sa ATP, ano ibig sabihin nun? The compounds here above ATP, they are used to produce ATP. So, huwag niyong kakalimutan itong mga compounds ito, especially phosphoenol pyruvate, 1,3-bisphosphoglycerate. Kasi babanggitin natin sila later sa glycolysis. No? They will be used to produce ATP. And then creatine phosphate, as I said earlier, in muscle physiology, it's useful for rapid release of energy. So, these compounds are used to produce ATP, while the compounds below are produced using ATP. Okay? The compounds below are produced using ATP. So, paano ba natin pinoproduce yung ATP? So, this is an important concept. There are two ways by which our body can produce ATP. And they are very different from each other. The first one is called substrate level phosphorylation. And the second one is called oxidative phosphorylation. So once again, ulitin natin. What are the two ways to produce ATP? Number one is substrate level phosphorylation. And number two, oxidative phosphorylation. Anong pinagkaiba nila? Between the two, our preferred method, yung mas maraming mapoproduce na ATP will be oxidative phosphorylation. So as much as possible, if we can try to produce ATP using oxidative phosphorylation, yun yung mas gusto natin because we produce a lot more ATP using oxidative phosphorylation. What's the disadvantage? It has two requirements. What are the two requirements? Number one, you need to have oxygen. And number two, the cell needs to have mitochondria. Meaning to say, if you don't have oxygen or if you don't have mitochondria, you cannot produce ATP via oxidative phosphorylation. Okay, sa kailangan both of them should be present. Kung kulang kahit isa, hindi na tayo pwedeng mag-oxidative phosphorylation. How will we produce ATP? We will produce ATP via substrate level phosphorylation instead. Okay? So what are the two requirements of oxidative phosphorylation? Number 1, oxygen. Number 2, mitochondria. And both have to be present. So, dito tayo sa substrate level phosphorylation first. Ano ibig sabihin ng substrate level phosphorylation? So, we use this to produce ATP when you don't have oxygen or when the cell does not have mitochondria. So, you see here, right? So, you see here, this is ADP. So, wala kang pera. No? Kaya nga ADP lang. So, wala kang pera. ADP lang. So, sa ADP, kulang ka ng pera. Wala kang ATP. So, remember, ATP, pera. Okay? So, si ADP, wala siyang pera. Naglalakad-lakad siya, nag-iisip siya, paano ba ako kukuha ng pera? Buti na lang, nakita niya si phosphoenol pyruvate. No? So, si phosphoenol pyruvate, kumpare niya yan. No? Si phosphoenol pyruvate, kumpare niya. Sabi niya, nakita niya si phosphoenol pyruvate. Sabi niya, Pards! Long time no see. Diba? Kita niya. Long time no see. Nag-hi siya. So, nakita naman siya ni phosphoenol pyruvate. Okay, magkaibigan naman sila. Tapos, ano gagawin ni ADP? Sasabihin niya kay ADP, kay phosphoenol pyruvate, Parts, tagal na natin din nagkita. Pautang naman, oh. Okay, so uutangan niya. Kasi nga wala siyang pera. So, ano sabi natin kanina? Si phosphoenol pyruvate, if you remember, si phosphoenol pyruvate ay mas mayaman, no? So, dahil mas mayaman siya, anong sasabihin niya? Sure, no? So, pahihiramin niya, no? So, yung phosphate ni phosphoenol pyruvate, ililipat natin kay ADP. So, that's what we call substrate level phosphorylation. So, yung phosphate, ililipat from phosphoenol pyruvate to ADP, and you now have ATP. Ganun lang kadali, no? Ganun lang kadali. Pautang. So, si ADP, nangutang ng phosphate from a high energy compound para maging ATP. May ATP na tayo, may pera na tayo. Okay? Remember, when do we do substrate level phosphorylation? When we don't have oxygen or when we don't have mitochondria. What about oxidative phosphorylation? So, oxidative phosphorylation is different. In oxidative phosphorylation, dadaan tayo sa electron transport chain and ATP synthase. No? So, you will see here, mamaya i-discuss natin to. Yung electron transport chain, no? This is where through a series of oxidation reduction reactions, the electrons flow through through complexes, no? So it flows to 4 complexes, eventually reaching water, and in the process, right, we generate a proton gradient that helps us synthesize ATP. So pupunta tayo sa factory or pagawaan ng pera. So gagawa tayo ng ATP from scratch. Okay? So, more on that later. Okay, so, once again, no? So, for now, ano lang kailangan niyong tandaan? The difference between substrate level phosphorylation and oxidative phosphorylation. So, substrate level phosphorylation, this is the definition. This is done through coupling reactions where a phosphate group is transferred to ADP from another substance with a higher delta G, no? Galing sa isang mas mayaman. So, there are 2 pathways where substrate level phosphorylation occurs. So, number 1 will be glycolysis, and number 2 will be the Krebs cycle. So, huwag niyong kakalimutan, no? So, glycolysis and Krebs cycle, this is where we undergo substrate level phosphorylation. The second way to produce ATP is oxidative phosphorylation. This is the greatest quantitative source of high-energy phosphate in aerobic organisms. Kaya nga sabi natin, di ba? Anong preferred method of ATP production? Oxidative phosphorylation. Okay, so, free energy comes from successive oxidation of substances in the respiratory chain within the mitochondria. And molecular oxygen is the final substance to be reduced. Kaya kailangan ng mitochondria, kailangan ng oxygen. Let me ask you a few questions. So, for example, muscle. If you have muscle tissue, no? well-oxygenated muscle. It's exercising. Nag-exercise yung biceps mo, no? No, nagba-bicep curls ka. So ideally, yung biceps mo, paano siya kukuha? Paano siya kukuha ng energy? How will it produce ATP? So ang gusto natin ay oxidative phosphorylation, 'di ba? May oxygen, may mitochondria. Pero kunyari, yung biceps mo nag-fatigue, no? Or for example, nakulangan ka ng oxygen, nag-asthma attack ka, nakulangan ka ng oxygen. Ano mangyayari dun sa muscle? Your muscle will shift to substrate level phosphorylation kasi nag-fatigue na siya naubusan na siya ng oxygen. So mag-shift siya to substrate level phosphorylation. How about your red blood cells? Paano kaya nagpo-produce ng ATP yung red blood cells? Your ATP is produced via substrate level phosphorylation. Why? Bakit ganun, sir? Marami naman siyang oxygen, di ba? Yung red blood cells nga nagdadala ng oxygen. But you must remember, your mature red blood cells do not have mitochondria. Kaya yung mature red blood cells, they will produce ATP only via substrate level phosphorylation. Okay? How about your neurons, your nerve cells? So your neurons in the brain, paano sila dapat magpo-produce ng ATP? So kung alive ka and well and healthy, your brain cells will produce ATP via oxidative phosphorylation. Paano kung bigla kang nag-stroke, no? So na-occlude yung internal carotid artery mo, no? Wala nang blood flow to the brain, you get a stroke, an ischemic stroke. Then, because of lack of oxygen, your brain cells, your neurons, they will produce ATP via substrate level phosphorylation only. And unfortunately, that will not be enough to meet the energy needs of the brain. Okay? So remember that. So just remember the differences between substrate level phosphorylation and oxidative phosphorylation. The next topic is electron transport chain in oxidative phosphorylation. So this is the second way by which we will produce ATP. First, let's review the anatomy of your mitochondria. So if you remember your mitochondria, anong tawag natin sa kanya sa biology? It's the powerhouse of the cell. So why do we call it the powerhouse of the cell? It's called the powerhouse of the cell because we produce a lot of ATP here. No, we produce a lot of ATP here. Now let's review the microscopic anatomy of your mitochondria. So this is your mitochondria. You must remember your mitochondria is an outer membrane and an inner membrane that's thrown into folds, called cristae. This is very important to understand, the difference between your outer membrane and your inner membrane. Bakit ganon? Bakit kailangan may outer membrane but kailangan may inner membrane? Now, inside, that's your mitochondrial matrix. So the mitochondrial matrix, it's inside the mitochondria. It will contain your enzymes, mitochondrial DNA, mitochondrial RNA, mitochondrial ribosomes. So, anong pinagkaiba ni outer membrane from your inner membrane? The outer membrane is freely permeable to most ions and small molecules. So, halos lahat pinapalusot niya. In contrast, the inner membrane is impermeable to most small ions. It's very selective. Konting-konti lang yung pinapalusot niya. It's very tightly regulated. Again, the outer membrane is freely permeable and the inner membrane is very selectively permeable. Why do we need cristae? The cristae will increase surface area, So para lang yung gyrosol sa and brain, so that there's more space, for your there's more areas for you. There are more areas for your enzymes or your electron transport chain. You must remember that because your mitochondria is a source of energy, if you have mitochondrial diseases, when you have mitochondrial diseases, sino kaya yung severely impacted? Which organs? It will be the organs that require a lot of energy, like your nervous system, your brain, nervous system, and your musculoskeletal system, diba? Because your muscles require a lot of energy. Okay? So, when we eat carbohydrates, fatty acids, proteins, and fats. So our diet consists mainly of carbohydrates, proteins, and fats. We break them down into monosaccharides, amino acids, fatty acids, cholesterol. And then we digest them. We catabolize them to produce carbon dioxide and water. When we digest it, so carbohydrates, fatty acids, amino acids, proteins, magiging carbon dioxide and water lahat yan. In the process, no, we generate what is known as electron carriers. So yung energy nung kinain natin, yung energy from the carbohydrates, proteins, and fats, lahat ng energy na yan, ita-transfer natin dun sa electron carriers. These two electron carriers are nicotinamide adenine dinucleotide and flavine adenine dinucleotide, NADH and FADH, and these were what we call electron carriers. Okay, so kanina sabi natin, when we see ATP, ano dapat maisip natin? Pag nakakita ng ATP, maisip natin pera, no? We must remember pera. Pero kapag nakakita naman tayo ng NADH tsaka FADH, anong gusto kong isipin nyo? Ang iisipin nyo, cheque. Again, pag ATP, pera. Pag NADH, FADH, cheque. What does this imply? Okay, for example ako, gusto kong pumunta sa Jollibee. Pwede ko bang ipangbayad yung cheque? Pwede ko ba yung sabihin, C3 nga po, tapos bibigay ko yung cheque? Hindi, di ba? Or for example, bibili ka ng red underwear sa SM para sa board exam. No? Pupunta ka sa SM, pwede mo bang ibigay yung cheque? Siyempre, hindi. Or for example, sumakit yung ulo mo, ang sakit ng ulo mo, punta ka sa Mercury Drug, pabili po ng Bioenergetics. Pwede mo bang ibayad yung cheque? Of course not. Now, what do you need to do? Kailangan yung cheque, papalitan muna natin into cash. No, saan natin pinapapalit into cash? Sa bangko, no? So parang ganun yung analogy na gagamitin natin. Whenever we digest food, no, we produce electron carriers, NADH and FADH, they contain energy. Pero hindi pa natin sila pwedeng gastusin. We need to bring them first to the bank. Yung bank, yun yung electron transport chain. At doon sa electron transport chain, yung cheque natin mako-convert into cash or ATP para pwede na natin gamiting panggastos. Okay? Pwede na natin silang gamiting panggastos. So the electron transport chain is the final common pathway by which electrons from the different fuels of the body flow to oxygen. So it is located in the mitochondria, specifically in the inner membrane. So ibig sabihin, if you don't have mitochondria, you don't have an ETC. There are two electron carriers used in the ETC, nicotinamide adenine dinucleotide, NAD, and flavine adenine dinucleotide or FAD. So you see here, no, FAD, no, and NAD, these are their structures. So NAD is derived from vitamin B3, no? NAD is derived from vitamin B3, whereas FAD is derived from vitamin B2. So NAD, no, nicotinamide adenine dinucleotide from V3 niacin, whereas flavine adenine dinucleotide is derived from vitamin B2 riboflavin. So huwag niyong kakalimutan yan, ha. So your electron carriers NAD and FAD. So they receive electrons. So kung si NAD plus saka si FAD, dagdagan natin sila ng electron. So remember, hindi tayo pwede magdagdag ng electron lang, no, kasi magiging negative yon. So si electron may kasama siyang proton, kaya nagiging NADH, no. So kaya nagiging NADH kasi si NAD plus bibigyan mo ng electron, pero si electron may kasamang proton, H. In contrast, no, so si FAD naman, FAD magiging FADH2. So what will happen in the ETC? So may dala-dala kang cheque. So ganting itsura ng electron transport chain. So you will see here, okay, this is the electron transport chain. So if you look here, no, this is the inner mitochondrial membrane. So that's the inner mitochondrial membrane. Anong sabi natin sa inner mitochondrial membrane? Your inner mitochondrial membrane is impermeable, or some would say very selectively permeable. You will see here your components. So these are your complexes, no? So you have complex 1, complex 2, complex 3, and complex 4. Okay, so complex 1, complex 2, complex 3, and complex 4. Now, anong iisipin natin? Ganito yung gusto kong isipin ninyo. So, kunyari, no? So, you will see here NADH and FADH. Kung ako, magpapapalit ako sa bangko, tapos ang hawak ko na cheque ay NADH. Okay, so you will see, no? May hawak ako cheque, NADH. Saan ako pupunta? Pupunta ako kay teller 1. Okay? Pupunta ako kay teller 1. Pero, kung ang cheque na dala ko ay FADH, kanino ako pupunta? Pupunta ako kay teller 2. Okay? Pupunta ako kay teller 2. Again, always remember this. There are 4 complexes in the electron transport chain. Where are they located? In the inner mitochondrial membrane. If you have NADH, NADH will enter the ETC via complex 1. If you have FADH, FADH will enter the electron transport chain via complex 2. So, what happens in the electron transport chain? No? Sa electron transport chain, no? It's a series of complexes that pass electrons until it is received by the final electron acceptor. Okay? Anong ibig sabihin nun? Ito kasi yung kwento dyan. So, sa bangko, no? Sa bangko, pagpasok natin, no? Pagpasok natin sa bangko, merong 4 na teller. Si teller 1, si teller 2, si teller 3, tsaka si teller 4. No? So, good morning. Papapalit po akong cheque. No? Pag ang hawak mong cheque complex 1 ay NADH, punta ka kay teller 1. Pero ito kasi yun. Si teller 1, no, tamad yan. No, tamad siya. No? So, pag nabot mo sa kanya yung cheque, kukunin niya yung NADH. No? Kukunin niya yung NADH. No? Tapos, kukunin niya yung electrons. No? Ang tatanggapin niya yung electrons. So, ang mangyayari, yung electrons mapupunta sa kanya, kunin niya yung cheque, si NADH magiging NAD ulit. So, yung cheque, anong gagawin niya sa cheque? Hindi niya yung papalitan into cash. Anong gagawin niya? Bibigay niya kay coenzyme Q. So, you see here, this is coenzyme Q. Bibigay niya kay coenzyme Q. Okay? Ano si coenzyme Q? Si coenzyme Q, sabi niya, no? Si coenzyme Q, hindi naman yan teller. Hindi naman ako teller. No? Ano ba si coenzyme Q? Si coenzyme Q, security guard yan. No? So, security guard. No? So, Q. Q, security guard. Hindi naman po ako teller. No? So, anong gagawin niya? Bibigay niya yan kay teller 3. Sabi niya kay teller 3, papalit mo daw. So, yung electron, ipapasa naman niya kay complex 3 or teller 3. Si teller 3, tamad ulit. No? Tamad ulit si teller. Tamad talaga itong mga teller na to. No? Anong gagawin niya? Ayoko nga. So, papasa niya kay cytochrome C na ipapasa kay teller 4. No? Si teller 4, sabi niya, naku, kawawa naman to. Kanina pa sa teller 1, pinagpapasa-pasahan. Anong gagawin niya? Finally, teller 4, no? Or complex 4, also known as cytochrome oxidase. Complex 4 cytochrome oxidase, yung electrons ipapasa niya kay oxygen. So you will see here, you have a half oxygen molecule. The half oxygen molecule will accept the electrons. And finally, oxygen becomes water. So in the process, no, from teller 1, no, pinasa-pasa natin yung electrons hanggang makarating kay oxygen, and oxygen becomes water. So that's why we call oxygen the final electron acceptor of the electron transport chain. Paano naman kung ang hawak mong cheque FADH? Paano naman kung ang hawak mong cheque ay FADH? So, may dala akong cheque FADH. Pupunta ako kay teller 2 kasi siya yung tagatanggap eh. Diba? So, ang gagawin ko, dala-dala akong cheque. Punta ako sa bangko. Okay? Papalit po into cash. So, si teller 2, ibibigay niya ngayon kay coenzyme Q. Tamad kasi si teller 2 eh. Papalit daw. Si coenzyme Q, sabi niya, hindi naman po ako teller. Security guard lang po ako. Bibigay niya ulit kay teller 3. Si teller 3, papasa na naman niya kay teller 4. And then si teller 4, ipapasa finally kay oxygen. Para maging water. Okay? Ito pa yung sasabihin ko. Ngayon, yung mga teller na yan, merong secret. Okay? Meron silang secret. take a look at teller 1, teller 3, and teller 4. Si teller 1, si teller 3, tsaka si teller 4, every time nakakahawak sila ng cheque. Okay? Again, teller 1, teller 3, teller 4, every time nakakahawak sila ng cheque, umuutot sila. So you see that? Nagpo-produce sila ng H+. Okay? So, they produce protons. No? So yung protons from the mitochondrial matrix, nilalabas nila into the intermembranous space. Kaya siya tinawag na intermembranous. This is the space between the outer membrane and the inner membrane. So ano ibig sabihin noon? So, ibig sabihin noon, no? Kunyari ang check mo ay NADH. Pag nagbigay ka ng NADH ETC, sino magpo-produce ng utot or protons? Si teller 1, si teller 3, tsaka si teller 4. Okay? Pero si teller 4, half lang, no? So, silent utot yan. So, half, 0.5, no? Tandaan nyo muna, 0.5 lang siya. Meaning to say, if you have NADH, you will produce how many protons in total? 1 plus 1 plus 0.5. 2.5 protons are pumped into the intermembranous space. 2.5 protons are pumped into the intermembranous space. How about if you have FADH? If you have FADH, now you will see here, if you have FADH, kung ang check mo FADH, ilan lang ang protons ang mapoproduce? 1.5 lang. Why? Because if you have FADH, dadaan ka kay teller 2, diretso na kay teller 3 tsaka teller 4. Hindi siya dumadaan kay teller 1. So you only produce 1.5 protons. And where is that produced? It's translocated into the intermembranous space. Okay? Intermembranous space. So yun muna yung tatandaan nyo. No? So ito, sa electron transport chain, binigay pa lang natin yung cheque. At pinagpasapasahan pa lang siya ng teller. Okay? Binigay pa lang natin sa bangko, tapos pinagpasapasahan siya ng teller. So what are the things you need to remember? No? So what you need to remember is this. So again, so you have 4 complexes. Complex 1 to 4. No? Complex 1 to 4, they are fixed to the intermembranous space. They are fixed in the inner mitochondrial membrane, except yung security guard, si coenzyme Q, no? And cytochrome C. So coenzyme Q is the only non-protein component of the ATC. No? So CoQ. So siguro na rin yun yung mga supplement na CoQ10, no? Ito yun, no? Yung mga heart vitamins daw. There are 3 tellers that produce protons. So complex 1, complex 3, and complex 4. Now, what happens, no? You produce a lot of protons. Habang nag-encash ng check, we produce a lot of protons in the intermembranous space. And then, the final electron acceptor is oxygen. What happens to oxygen? Oxygen becomes water. So, paano yung naging ATP? No? So, you see here, if this is your ATC. So again, look, it's called the intermembranous space because you have the outer membrane and the inner membrane. So what happens? So NADH and FADH, papasa natin yung cheque. No? You translocate protons, 1, 2, and 0.5. No, habang nag-encash ng check, what happens? The intermembranous space becomes more positive. Okay? The intermembranous space becomes more positive. Okay? Kasi dumadami yung protons, dumadami yung H+. It becomes more acidic. So, it becomes more positive, it becomes more acidic relative to the mitochondrial matrix. So, paano yun magiging ATP? Doon papasok si complex 5. So, you see here complex 5 on the left side. No? So, si complex 5, yung mga protons, ayaw nila sa intermembranous space. They do not like it there. Ang gusto nila, bumalik sa mitochondrial matrix. So the protons, can you imagine, habang nagpapa-encash ng check, dumadami yung protons dito, pero sabi nila, ayaw namin dito. Gusto namin bumalik. Bakit hindi na lang sila lumusot sa inner membrane kung gusto pala nilang bumalik? Because the inner membrane is impermeable. No, it's impermeable. So, ano mangyayari? Buti na lang nandun si complex 5. So, this is complex 5. Si complex 5, makita niya yung mga protons. Uy, gusto nilang bumalik. Anong gagawin niya? Palulusitin niya yung mga protons pabalik. Pero may catch, no? So, palulusutin niya yung mga protons, pero ganitong nangyayari dyan, no? Pag lumusot yung mga protons, no? So, you will see here, lulusot yung proton dito. Okay? Ito, umiikot to. No? So, itong part na to, umiikot siya. Okay? Every time may dumadaan na proton, iikot siya. Ting! Okay? You've just produced ATP. Ulit. So, pag may proton gustong bumalik, dadaan yung proton dito. Parang yung sa no, pag pumapasok ka ng LRT, MRT, no? Pag, yung dating way, no? Yung dati, yung luma, nung hindi ko alam kung inabot nyo pa yun, eh. Nung pag pag lumusod yung proton, no? Pag lumusod yung proton, iikot to. Ting! In here, what happens? ADP is phosphorylated to become ATP. Nakuha. So, lulusot yung proton. Ting! You've just produced ATP. Ano ibig sabihin nun? So, kung yung check natin NADH, if you began with NADH, meron kang NADH, ilang proton yung mapoproduce? Ilang proton yung lulusot? 1, 1.5. So, ibig sabihin, 2.5 yung kailangan bumalik sa matrix. So, ilang ATP yung mapoproduce pag meron kang NADH? Okay? 2.5. Ting, ting, ting. 2.5 ATPs kasi yung babalik. Pero pag FADH yung check mo, ilang ATP mo mapoproduce? 1.5 lang. Ting, ting. Kasi nga, no, 1.5 protons lang yung na-generate natin. So, take note, ito yung ETC, electron transport chain, complex 1 to 4. It is coupled with oxidative phosphorylation, which happens in ATCP synthase, or complex 5. So, ito naman yun, no? So, you will see here. This is another illustration. Anong sabi natin? If you have NADH and FADH, ipapasa natin sa kanila yung check natin, no? So, NADH po, dadali natin sa electron transport chain. We translocate protons in the intermembranous space. 1, 1.5, no? So, yun yung ratio. So, 1, 1.5. Gusto ba nila doon? Ayaw nila doon. Kailangan nilang bumalik. Paano sila babalik? Babalik sila through complex 5. So, you see there, lulusot yung mga protons. Paglusot ng mga protons, ting! And now we've produced ATP. So, that is what you call the Mitchell hypothesis. So, the Mitchell hypothesis is the chemiosmotic hypothesis that explains how free energy generated by the transport of electrons by the electron transport chain is used to produce ATP from ADP and phosphate. Okay, so, anong nangyayari? Again, you create an electrical and pH gradient. Sabi natin, the inner membranous space becomes more positive because it also has, and it becomes more acidic because it now contains more protons. Now, the protons have to go back through complex 5, resulting in the synthesis of ATP. Okay? So, tatandaan ha. So, para ka lang may cheque. Yung cheque, dadali mo sa bangko, found in the mitochondria, sa bangko, pasa-pasa sila kasi tamad yung mga teller, uutot yung mga teller, eventually yung electrons mapapas kay oxygen para maging water, yung intermembranous space magiging positive, ayaw nila doon, babalik sila, saan sila dadaan? Complex 5. At pag dumaan sila sa complex 5, ting! You've just produced ATP. Paano ngayon pag wala tayong oxygen? What if we do not have oxygen, as in cases of tissue hypoxia? So, for example, di ba, stroke. No, kunyari na stroke ka. O kaya na myocardial infarction. Pag sinabi natin stroke, the blood vessels that supply a brain all of a sudden, nawalan sila ng oxygen. Maybe from a blood clot. Okay? Or for example, hemorrhage. Pumutok yung blood vessel na nagsusupply sa kanya. Okay? So, the tissues become ischemic. Or in myocardial infarction, yung coronary arteries nag-occlude. No? So, yung myocardium, mawawalan siya ngayon ng oxygen. Okay? So, when there's lack of, kung walang oxygen, wala ngayon tayong final electron acceptor. So kung walang final electron acceptor, ano mangyayari? Magtatrabaho ba yung mga teller? Of course not. No? So hindi sila magtatrabaho. No? So pag wala ngayon yun, hindi na magtatrabaho yung mga teller. No? Titigil yung electron transport chain. So wala na tayong ATC and oxidative phosphorylation. And how will we now produce ATP? If we cannot produce ATP via oxidative phosphorylation, we will now produce it via substrate level phosphorylation. O, di ba? Huwag niyong kakalimutan yon. Sabi natin, yung substrate level phosphorylation, anong ginagawa natin doon? Pards! Pautang. No? Mangungutang tayo ng phosphate. Kanina tayo mangungutang? Sa mas mayaman. So, we will now be forced. Mafo-force tayo. Instead na magpo-produce tayo ng ATP, mafo-force tayo ngayon mangutang ng phosphate para lang makaproduce ng ATP. And, unfortunately, for highly aerobic tissues like neurons and muscle tissue, that's not enough. No? That's why we do not want tissue hypoxia. This will lead to cell death. Now, what you need to remember is this. We can inhibit the electron transport chain. So, some compounds, what they do is they block or they inhibit components of the electron transport chain. So pag inhibit natin yan, pag pinigilan natin yung mga teller, no? Pag pinigilan natin yung mga teller, hindi ngayon tatakbo yung electron transport chain. So this table is important, no? You need to know this. So you will see, may mga pangalan yung mga teller natin. Complex 1, complex 2, complex 3, and complex 4. Complex 1 is also known as NADH dehydrogenase. Complex 2 is succinate dehydrogenase. Complex 3 is your cytochrome BC1 complex. And finally, pinakasikat, complex 4, cytochrome oxidase. So, you will see there, may kanya-kanya silang kalaban. May kanya-kanya silang inhibitor. And what does this imply? So, for example, if I give malonate, what will happen? Malonate will inhibit complex 2. Titigil si complex 2. No? Hindi na siya tatanggap ng cheque. Paano na tayo magpo-produce ng ATP? Ang mangyayari, hindi na tayo pwede magpa-encash ng FADH. Makakapagpa-encash na lang tayo ng NADH through complex 1. Okay? O how about if we give cyanide? If we give cyanide, ano mangyayari? The electrons will not be able to flow to oxygen anymore. Kasi wala na magpapasa ng electrons kay oxygen. Kaya ayaw natin yon. So for the board exam, no, what they tend to ask is they ask you, no, which complex is inhibited. So for example, if you give amytal, which complex? Complex 1. No, sometimes they ask ang nasa choices yung mga real names. So that's why you need to know this, no? Alam mo na yung NADH dehydrogenase, yun yung complex 1. So you see here, so for example here, if we give, no, so if we give amytal, no, amobarbital rotenone, what will happen? They will inhibit complex 1. Hindi na tayo makakapag-produce ng ATP from NADH. Makakapag-produce na lang tayo from ATP from FADH. What about if we give malonate? Then we will stop complex 2. Again, hindi na naman tayo makakapag-produce naman from FADH via NADH lang. Ang pinaka-severe, no? Ang pinaka-ayaw natin is if we stop complex 3 and complex 4. Kasi pag in-stop mo yung complex 3, so if you give, no, antimycin A or dimercaprol, you stop complex 3. Or if you give cyanide, hydrogen sulfide, or carbon monoxide, ito yung mga pinakasikat, complex 4, no? So pag hindi nyo maalala ang sagot, complex 4, no? Kasi yan yung mga sikat. Cyanide, hydrogen sulfide, carbon monoxide. Bakit mas severe yung effect kapag complex 3 and complex 4 yung binlock natin? Kasi wala silang alternate, di ba? Si complex 1 at si complex 2, alternate sila ng isa't isa. Kaya pag binlock mo yung sila doon, pwede pang gumawa ng ATP via the other pathway. Pero si complex 3 and complex 4, wala yung kapalit. That's why complex 3 and complex 4, pwede yung gamiting weapons of mass destruction. No? Pwede silang chemical weapons. They are poisons. What will happen? If you give ETC inhibitors, you will see here, they will prevent the passage of electrons, no, by stopping components of the electron transport chain. The redox reactions will stop. No? The redox reactions will stop. So, yung reduction, no, mawawala. The effects. So what are the effects? You will decrease oxygen consumption. Bakit? Dahil hindi tumatakbo yung ETC, no? Wala nag-encash. Si oxygen nakatenga lang doon, naghihintay lang siya. Naghihintay lang siya ng dadating na electron na hindi naman dadating. Number 2, increased NADH, NAD, and FADH ratio. So tataas yung NADH, tataas yung FADH relative to NAD and FAD respectively. Kasi nga hindi sila na-encash. And finally, there will be decreased ATP kasi kung wala nag-encash, no? Di wala tayong napoproduce na ATP. The other group of compounds that decrease ATP production are what you call your uncouplers. So what are your uncouplers? So your uncouplers, they increase the permeability of the inner mitochondrial membrane to protons. I'll try to think, anong effect nun? So kanina sabi natin, di ba? Sabi natin, yung mga protons, gusto ba nila sa intermembranous space? No, ayaw nila doon, no? So for example here, if you look, ito, ayaw nila dyan. Gusto nilang bumalik at bumabalik sila via complex 5. Anong ginagawa ng uncouplers? Yung uncouplers, gagawin nilang permeable yung inner membrane. So pag ginawa nating permeable yung inner membrane, ano ngayon yung effect nun? Hindi na sila mapipilitang dumaan kay complex 5. Sabi na, uy, nakakalusot naman pala tayo dito eh. Bakit pa tayo dadaan kay complex 5? Bakit problem yon? Problem yon kasi pag hindi na sila dumaan kay complex 5, wala na tayong ting, diba? Hindi na tayo gagawa ng ATP. So, encash tayo ng encash, pero walang napoproduce na ATP. So, the ATC keeps running. So, that's why, no, what happens, no, no, ang effects niya, you will see here, increase oxygen consumption, no, tanggap ng si oxygen ng electrons, no, bumababa yung level ng cheque, NADH, but no ATP is produced. We do not produce ATP. Instead of producing ATP, we generate heat. So instead of producing ATP, you generate heat. So you need to memorize this. There are 3 uncouplers. No? Itong tatlong uncouplers na to, kailangan lang kabisado niya sila. 2,4-dinitrophenol, aspirin, and thermogenin. So yung aspirin, no? Aspirin overdose. When you overdose with aspirin, you will present with metabolic acidosis. Kasi salicylic acid dyan, no, mag-accumulate sa blood. At the same time, patients will become hyperthermic. So tatandaan niyo yan, ha? If you are presented a case, tapos meron siyang metabolic acidosis, tapos hyperthermia, ang iisipin nyo aspirin toxicity. Kasi nga, aspirin acts as an uncoupler. How about brown fat? Yung brown fat, thermogenin or brown fat, your infants, no? Especially your neonates, hindi pa sila marunong mag-shiver, no? So we shiver. Why do we shiver? We shiver to generate heat. Now your neonates, they cannot shiver. Hindi pa sila marunong. So how do they generate heat? How do they keep themselves warm? By burning brown fat or thermogenin. How about yung malignant hyperthermia, no? Ano naman yung malignant hyperthermia? Yung malignant hyperthermia, sometimes inhalational anesthetics, no? They can trigger a reaction in susceptible people, no? So nagkakaroon ng uncoupler effect, no? So there will be a marked rise in temperature and respiratory acidosis. Okay, so tatandaan lang, no? So uncouplers, 2,4-dinitrophenol, aspirin, and thermogenin or brown fat. ATP synthase inhibitors, nag-iisa lang to. Oligomycin, no? So tandaan niyo yung O, no, oligomycin, yung umiikot sa ATP synthase, no. So ang yung letter O, yung bilog, ang mag-inhibit sa kanya letter O, oligomycin. If you block ATP synthase, so you see that? If you block ATP synthase, syempre, di walang dadaanan ngayon yung mga protons. At dahil walang dadaan yung mga protons, titigil na din yung ETC. Sasabihin nila, uy mga parts, wala naman tayong dadaanan dito, no? Tigil nyo na yan. Tigil nyo na yung pagpapa-encash kasi nga barato. Okay? Kasi nga barato. So remember, oligomycin, it's a direct inhibitor of ATP synthase. Finally, atractyloside. Atractyloside also decreases ATP production. How? So, di ba sabi natin sa complex 5, gumagawa tayo ting, ting, ting ng ATP. Para siyang factory ng ATP. Kailangan din niya ng raw materials. So, there's constant transport of ATP and ATP in and out of the mitochondria. So, when you give atractyloside, this transport in and out the mitochondrion is blocked. No? This transport is blocked. Okay? So, once again, anong important dito? You need to remember that these three groups, ang effect nila lahat pare-pareho. They decrease ATP. Pero iba-iba sila ng mechanism. So, ATC inhibitors, they will block either complex 1, 2, 3, 4. You just need to memorize. Sino ba kay complex 1? Sino ba kay complex 2? Sino ba kay complex 3? Sino ba kay complex 4? Sinong pinakaayaw natin? Complex 3 and 4. Sino yung mga pinakasikat? Complex 4, no? Cyanide, hydrogen sulfide, and carbon monoxide. They block complex 4. Kaya, no? Pag may cyanide poisoning, you have cyanide poisoning, delikado yun. Hindi ka magpo-produce ng ATP via oxidative phosphorylation. Kasi nga, binlock natin si complex 4. And then, your uncouplers. The uncouplers, they increase the permeability of the inner membrane. Tuloy-tuloy yung ETC, but you don't produce ATP. Instead, you generate heat. Your direct ATP synthase inhibitor, nag-iisa lang yan, oligomycin. And then your ADP-ATP transport inhibitor, your atractyloside. What are reactive oxygen species? So yung reactive oxygen species na tinatawag, remember the role of oxygen. The role of oxygen, it's the final electron acceptor, wherein you have a half electron molecule. No? So you will see here, if you look at the diagram, merong naghihintay dito na half oxygen molecule. Siya ngayon yung tatanggap ng electron para maging water. So akala mo, ang galing naman niya, tinatanggap niya yung electron para maging water. Thank you, no? But actually, this can be very damaging to the tissues. The problem with the half oxygen molecule, although maganda siya kasi nakakatulong siyang gumawa sa atin ng ATP, no? It generates unstable products. No? Ito yung mga tinatawag na free radicals. So the half oxygen molecule generates free radicals such as superoxide, hydrogen peroxide, and hydroxyl radicals. We can use them, no? Especially your neutrophils use them to kill bacteria. But sometimes, they can cause tissue damage. So, meron tayong tinatawag na reperfusion injury. Ano ibig sabihin nun? So, for example, yung pasyente na stroke. So, nagsarado, nabarahan yung middle cerebral artery niya. No, merong blood clot, nasarahan yung middle cerebral artery. Buti na lang, nadala siya dun sa hospital capable of thrombectomy. Ibig sabihin, no, magpapasok ng wire to the brain, tapos i-retrieve yung blood clot. So, pag natanggal na yung blood clot, biglang papasok ngayon ulit yung blood into the brain. Shhh! Mape-perfuse na naman yung brain. Unfortunately, that can be dangerous. Why? Kasi from complete stop, bigla mong patatakbuhin yung electron transport chain. At pag bigla mo siyang pinatakbo, dadami yung free radicals. Dadami yung free radicals natin. At pag dumami yung free radicals, this can cause tissue damage. The same can happen for reperfusion following thrombectomy in myocardial infarction. No? So, di ba yung mga nag-MI, no? Tapos ng thrombectomy, natanggal nila yung blood clot. No? Na-restore nila yung blood flow sa heart. They can also sustain reperfusion injury. So, to protect ourselves, no, we have these defenses against reactive oxygen species accumulation. So you have different enzymes. So you have your catalase, peroxidase, and superoxide dismutase. So catalase, for example, converts hydrogen peroxide to water and oxygen. Okay, so take note, even though reactive oxygen species can be used to kill bacteria, they can be damaging to the tissues. And you generate them in the electron transport chain. So in summary for this topic, no, remember, bioenergetics, spontaneous reactions have negative delta G, indicating loss of free energy. So remember, our reactions, if you want them to be spontaneous and favorable, you have to have a negative delta G. How do you compute delta G? So pwedeng delta G of the product. So energy level of the product minus energy level of the substrate. If the result is positive, it's an endergonic reaction. It is not spontaneous. It is not favorable. In contrast, if delta G is negative, that reaction is exergonic. It is spontaneous. It is favorable. How do we compute delta G using enthalpy and entropy? Delta G is equal to delta H minus T delta S, where T is the temperature in Kelvin. Next, adenosine triphosphate is the energy currency of the cell. Remember the role of ATP? Whenever you hydrolyze ATP, you release 7,300 calories. ATP is produced either by oxidative phosphorylation or substrate level phosphorylation. What's our preferred method of ATP production? Oxidative phosphorylation. However, what are the two requirements? Oxygen and mitochondria. If you don't have oxygen or if you don't have mitochondria, how will you produce ATP? Via substrate level phosphorylation. At yung substrate level phosphorylation ay transfer of phosphate from a higher energy compound. So, pautang galing sa mas mayaman. Finally, the electron transport chain generates a proton gradient that drives ATP synthesis. So, produce ATP via oxidative phosphorylation, we need the electron transport chain, yung bangko, where we will bring NADH and FADH. NADH yields how many ATPs? 2.5. How about FADH? 1.5. Inhibitors of the respiratory chain cause depletion of ATP and may be fatal. So just memorize which are ETC inhibitors, which are uncouplers, which is the direct ATP synthase inhibitor, and which blocks the transport of ADP and ATP. Thank you for listening to this lecture.